So welcome to lecture 16. Coming down the back side of classes. So this week in lab, we will check our silos and do our first balanced rations as a class. Next exam, taking a vote on that. I have it tentatively scheduled for the 9th, but I'm flexible on that. So we've got Monday the 7th, Wednesday the 9th, Monday the 14th, or Wednesday the 16th seems appropriate. We're going to cover 9 lectures. So the last lecture on the exam will be that Monday the 31st. So you guys want some time to think about it. We do a final vote next class. Check your schedules. We got 1 nod yes, 2 nods. Okay, so check your schedules. So the first or second Monday and Wednesday of the month. So you'd either have a week to study or 2.5 weeks to study, depending on what you're going through. So we'll do that vote next time. Rest of the semester kind of looks like this. I should get that posted on Blackboard. We're going to go through proteins, dry cow rations in lab this week. We're going to minerals. We got our lab practical part 1, and then minerals, vitamins, spreadsheet balancing for lactating dairy cows. And we'll spend the last few lectures talking about feeding cows. So taking what we've learned and putting it in a practical setting. And then your last exams are there at the end. So, Brittany, we need to find you another spot. Or you can do both of them on that Thursday. Do you have a preference? So Brittany, unfortunately, was one of the last to sign up, and she only had that spot available. It's hard for her to double herself in that area. But we can make that happen or adjust that. Does anybody want to trade moving down to the 10th or up to the 10th and allowing Brittany to take your spot? Take a look at that. We'll make some decisions on Wednesday. So, question, your next question, I believe, is due today. If you haven't turned in your 3rd question, it might be due today. So give that some thought. Remember that questions about the class content as well as the class discussion for management and all that stuff, those are all valid questions one way or the other. So lab practical, we talked about that. Next week, we'll probably sign up for our spots. We'll have 2 people at each time slot. So give some thought as to when you want to take that feed ID exam in a week from Thursday. Lab practical at the end. So we're going to discuss proteins. Anybody know anything about protein? Could be used for energy. Yep. That's the basis of the Atkins diets and the paleo diets and all that stuff. High protein diet where you're burning protein for energy as opposed to just carbohydrates. Anything else about protein? Muscles are made of protein. Most tissues are made of protein. So amino acids. So proteins are a string of amino acids stuck together. Ultimately, that's what we care about, but we'll show you why that's difficult to get a handle on with the ruminant. As I think I've mentioned before, the pig and chicken people laugh at us because we still use crude protein. They balance on an amino acid level. They're way ahead of us, and so we'll talk about some of those reasons here. So what is the key difference between carbohydrates, fats, and proteins? That is the presence of the element nitrogen. So none of those carbohydrates or fats have nitrogen in them. And that nitrogen is what we use to set those things apart. That's more often not how we measure things. So what sets things apart? The element or atom in protein is the nitrogen atom, specifically part of what's called the ammonia group. Amino acid has a acid on one end and an ammonia with that nitrogen on the other. So when we're dealing with our cows, we're usually looking at crude protein, and that is figured out or calculated by, we measure the nitrogen in the feed. We assume all the nitrogen in the feed is related to protein. That's a pretty good assumption, but not a perfect assumption. And then we know that on average, cow feeds are about 16 cow proteins that they consume are about 16% nitrogen. So I can measure the amount of nitrogen. I multiply it by 6.25, 6.25, which is what I would multiply 16 by to get 100. 6.25. So that amount of nitrogen, I multiply by 6.25. That represents the entire organic atoms associated with the protein. So we call that crude protein. And the reason it's crude is not all of that nitrogen is perfectly balanced at 16% of the total protein. There are nitrogen containing compounds like DNA that have aren't really proteins. So the word crude is used in this sense as a rough measure, a rough approximation. So we're getting close to it, but we are not perfect. If you look at your milk checks, it used to be crude protein is what you got paid for. They've since switched over to a more refined measurement, true protein, and they changed the coefficient. They're multiplying the nitrogen by 6. I think it's 632 to get the proper amount of total protein based on the nitrogen. So it's a crude example, and one of the things that you'll see is some weird things happen when you're measuring just nitrogen and multiplying by 6.25. Urea, which a cow can use to make protein, it's useful to the cow. It wouldn't be useful to the pig or the chicken, but we can put that nitrogen in a little taste of it. You give her too much, you'll kill her, but a little taste of it in there, and that helps the rumen microbes do more, generate more. The urea is a carbon with 2 ammonia groups stuck to it. So on a percent basis, it's 46% nitrogen. When we're balancing rations and putting urea in, the crude protein value for urea is 282% crude protein. So it's got a much higher amount there. It's not real protein, but with our cows, that 46% nitrogen is useful. So the atoms in protein, you're going to have your carbon, hydrogen, oxygen is your base. We stick a nitrogen or ammonia group on the end. You'll also have traces of sulfur, methionine, cysteine, a phosphorus now and again, and you'll see that in some of the pictures we have here. But dominated by the top three there, or top three lines, top four elements, and nitrogen is the defining characteristic. So we look at crude protein as a percent of the feed dry matter. We introduced that concept last week in lab. Alfalfa silage, it's not unusual for pure alfalfa stand to have a crude protein of 22%. Fairly typical average. Our alfalfa hay, if we do a good job with it, usually or typically or average, just a little bit lower. Does anybody have a guess of why that might be? Fermentation is going to play a factor. So, but we're putting up silage wet, we're probably not going to lose as many leaves as we would with hay. So that loss of leaves is going to bring down that nitrogen or protein amount just a little. Paired with legumes, we go from that medium crude protein down to that low protein. The mixed grass hay, so lots of different grasses together, generally lower protein. Corn silage, my brain always goes to 10% crude protein. If I don't know anything about the corn silage, I'll just default to 10%. A better number would be 9%. That's the average that has been submitted by thousands and thousands of samples. We come up with 9% crude protein. Corn itself is about the same protein percentage as the entire plant. So the grain is equal to the stalk. So in that 8, 9, 10 region. We talked about fractionating that corn grain, where we're trying to get that starch for human consumption. High fructose corn syrup and all other things that we might use that starch for. That wet milling process, most of the protein goes out at the corn gluten meal step. So most of the protein goes there. Our corn gluten feed, what's the characteristic or what do we know about that? Why do we feed that? What would be a good answer for corn gluten feed? If protein is the reason we feed corn gluten meal, why would we feed corn gluten feed? F is for fiber. So that's, if we needed some NDF, we needed some slowly fermentable stuff, bringing in that small particle fiber. That's a way to get that with the corn gluten feed. Beet pulp, pretty low. The whole beet is probably like 2% or 1% crude protein because it's all about the storage of starch in the sugar beets. Whole cottonseed, somewhere in that medium range, 24%. High moisture shelled corn, again, not that much different from our corn grain, that 8, 9, 10 region for corn. Barley, a little less starch, so it's got a little more protein. So, but it's still in that low category. All our cereal grains are generally low in crude protein. Because corn has the most starch, it dilutes out the protein and makes it a lower percentage of the dry matter. Soy hulls, the outside of the pod of the plant, pretty low. There's no reason to have a lot of protein in them. Soybean meal, we take the soybeans, we extract the fat. We're left with a product that's very high in crude protein. Usually say 53.5, but I rounded it to 54. So it's called soy 48 crude protein, but it says 54 up there. Can anybody guess why there's that discrepancy? So 54 on a dry matter basis, on an as-fed basis, it's 48. Then you'll have soy 44, they add some hulls in there, dilute it out. You'll drop down to 44% on an as-fed basis for crude protein. So crude protein needs for our animals, we're thinking in pounds per day. We have a lot. This is one of the ingredients that we need to focus on when we're feeding our cows, making sure we get our rations right. Making some errors with the smaller inclusion ingredients, a cow can make it up, but if we do a 10% error here, there's going to be some issues. So making sure we get the balance of carbohydrates and proteins right becomes a daily objective on our farms. And proteins, and what we really care about, are the building blocks of proteins. Proteins are a chain of amino acids strung together. Protein purposes, almost everything in the body's got protein in some way. So we got structures, bones, muscles, the structures inside the cell, the microskeletons in the cell are all made of protein. Enzymes, the machinery that gets all the things done that need to happen in the body, is another purpose for proteins. And another thing we'll look at is packaging. So we'll create this sphere of protein, and in particular, we'll put fats inside them. But we also can carry minerals, vitamins, can also be carried by protein carriers through the blood. So that allows them to move through the blood without sticking to the side or causing damage. The copper and iron carriers, those minerals are pretty nasty. Putting them inside that protein protects the body. If we have fat, fat's not going to flow through the blood real well. It doesn't mix with water. It's kind of like Italian dressing. The oil floats to the top. But if we stick it in a package, in a protein package, we can move it around the body quite efficiently. So we're looking at our protein. Plants can synthesize all their own amino acids from scratch. So we're looking at nature, about 20 amino acids that we need to make all the proteins that we have. Plants can synthesize their own amino acids. Why would that be helpful? That's certainly helpful to us as farmers. Yeah, we take the plants there, they create that protein, they create those amino acids from simple inputs. But if you're a plant, it's hard to walk down to the grocery store and get the amino acids you need. They have to be there. So the ability to absorb nitrogen and generate carbon skeletons from photosynthesis, stick those two things together to make proteins, is very necessary if you can't move. Microorganisms can synthesize their amino acids from scratch. So they don't need much protein. They're very, very small. So, and again, their ability to move is kind of limited. I would imagine if a microbe moved a half an inch, of course, over the course of its lifetime in the soil or something like that, that would be a huge trip. That's different than floating along in a river or being in a manure pit that's being agitated, but their ability to move is kind of limited. With our animals, their ability to move trades off for the ability to synthesize. So they can't synthesize all of them. So therefore, there's that dietary requirement we talked about when we had the 50 different nutrients. There are 20 amino acids. The body can synthesize 10. There are 10 that need to be provided in the diet. So we call those 10 amino acids that the animal cannot synthesize in adequate amounts. So the liver's not keeping up. We call them essential amino acids. So that cannot be synthesized at a rate which would provide normal growth or normal anything. So the essential means it needs to get to the small intestines and be absorbed. For you and I, that means we need to consume them. If we feed cow amino acids, does anybody know what happens to them first? Where do they go? Where's feed go first? Cow makes a mouthful of feed, chews it, swallows it. Where does it go first? Stomach, which compartment? Rumen, reticulum, what happens there? Fermentation, which is controlled by what? Microbes. The microbes have first crack at the amino acids, and they will change them, alter them, destroy them, make them. So because of the rumen, what we put in her mouth from a protein standpoint is not what ends up at the small intestine. So that's why I say whatever we feed the cow, the microbes mix it up. Whatever leaves that forestomach, rumen, reticulum, omasum, has to have those essential amino acids at the small intestines for that animal to absorb. And you can imagine trying to figure out what the microbes are doing is pretty complicated. What are they touching? What are they not touching? What are they creating? What are they destroying? That's a huge problem. The non-essential amino acids, we still need them in the body, but the liver can make them. So they're still essential from a dietary need, but they're not essential that we get them to the small intestines so they can be absorbed. And we talked about the limiting nutrient. Limiting amino acid is where we see this come into play most on dairy farms. Which amino acid is limiting that cow's production? Is it methionine? Is it lysine? Is it histidine? Is it arginine? We tend to feed extra methionine to our cows, extra lysine to our cows in an attempt to make sure that we've satisfied the amino acid need for those animals. So lysine, methionine, tryptophan are most likely limiting in our monogastric animals, chickens, pigs. They seem to be the least present. One of the things about a good corn-soybean mix, soybeans tend to be high in lysine. Corn tends to be high in methionine, so mixing those two grains together, we tend to make those less possibly limiting to the animal. So we're looking at our proteins. We have 20 more or less that are needed in our dairy cows. There's some weird amino acids when you get down to the vents underneath the ocean. Those creatures do some weird things, but 20 is where we'll stick. We mentioned already 10 essential, 10 non-essential amino acids. They all have the same basic blueprint. They have what's called a carboxyl end, which is an end of a molecule that's got an amino acid. You have a middle carbon, and then middle carbon has what's called an R group attached to it. And then you have the nitrogen end or the amino end. So every amino acid is carbon, carbon, nitrogen stuck together in a chain. Then you have this R group coming off the side. So what's different about the 20 different amino acids is what the R group is. Sometimes that could be as small as a hydrogen, just a single hydrogen is the R group. Other times you've got two big ring structures stuck together, a huge addition on the R side coming off that middle carbon. So if we look at that graphically, it looks something like this. So we have a common backbone for our amino acid. So every amino acid has that same structure. And then we have whatever's sticking off the carbon in the middle gives that amino acid's individual character. What happens with proteins, we put this chain together. So you've got that chain. Every red thing up there is a bond that's between the amino acids. You've got the R group sticking off. The downside of this particular diagram is you don't see the different sizes of the R groups in this particular picture. So the squares are around the individual amino acids. We're stuck together in a chain. So you're going to have thousands and thousands of amino acids stuck together to make certain proteins. Then other proteins like oxytocin, you only have 9. I think it's 9 amino acids in a row that make that. So you can have thousands and thousands in the chain. That common backbone flows through every protein, and its character comes from those R groups sticking off the side. So when we put those amino acids together, we have what's called a dehydration reaction, where you've got the OH group from the carboxyl end getting together with the amino group on the other one. Water falls out, and you have a peptide bond that links the two amino acids together. So this is a discussion of, or a presentation of all the different R groups that you'll find in nature. So you got the 20 different ones here. You don't have the color there, but the little open circles are hydrogen, the black dots are carbon, you got the reds are oxygen, blues are nitrogen, and yellow here and here is sulfur. So we got the simplest amino acid where the R group is just a hydrogen, glycine up top there. We have two different real categories. They either don't like water, or they like water. So if this group doesn't like water, this group does. What do you notice about that, just from an observational standpoint? What's the difference between those two amino acid groups? What about color? There's more red in that middle group. There's blue in that middle group. There's a little bit of yellow. So that top group doesn't like water. It's all carbons and hydrogens. We have charges in the middle here. Charges tend to create things that like water on some level. So you have R groups that like water, they'll turn the molecule toward water. And you've got R groups that don't like water, they'll tend to try to group together. And depending on which way the R groups twist around that common axis determines the shape of the protein in the saline solution that is the body fluid. So we'll spend more time on that in A and P. So you're seeing mostly black and white on that section. We got a lot more color down here, the sulfur, the nitrogen. The oxygen will result in unequal sharing of electrons, which will lead to a charge. So we have your non-essential amino acids. So there's 12 here. That's because some are sort of redundant. These are all the ones that can be made by the liver. It's great if we can supply them in the body. We don't need to. The liver, if it's got enough building blocks, can get the job done. But if we do provide them in the acid, that's one less thing the liver has to do, one less thing the liver has to make. What I want to focus, so made by the body or the cells, mostly the liver. What I want to focus on is the essential amino acids. These are the ones that cannot be produced at a sufficient rate, and they must be absorbed. We need them to be entering the body from the intestine, going to the liver, partitioned out through the body. We have enough. So you need to be able to recognize that these are the essential ones. So we have arginine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. As a general rule, the R groups here are bigger, so they're more complicated. So having the plants and the microbes make them as opposed to the cow's liver is going to save time and energy. The ability to walk around, select a diet will help make sure they're provided in the diet. Usually with our dairy cows, our lactating dairy cows, we're talking about supplementing lysine and methionine. Protected lysine, protected methionine. Has anybody heard of those before? So the idea of protection is that they go in the rumen and the microbes can't touch them. Eventually they'll funnel out, sieve out. They get to the abomasum, they get hit with acid and they're freed up. They move to the small intestines and they're absorbed. So if you've got a good feeding program, a good nitrogen program, everything's kind of balanced out roughly right, the addition of those protected amino acids usually will give you a response. But if we don't have a lot of other things right first, we usually don't get a response. So you got to make sure everything's lined up so that lysine, methionine, you know they're limiting. When you know they're limiting, there can be a response to them. But if you're limiting on energy or carbohydrates, minerals, vitamins already, adding lysine and methionine to the diet is not going to get you a response. So I want you to be able to recognize essential amino acids. So if somebody comes and sells you on your farm, I've got the amino acid that's going to work for you. You need to recognize that, yes, it's necessary. No, it's not. So I've got a glycine amino acid supplement that you really, really need. They're lying to you. So there's a couple different ways that you can recognize them. You can go through and memorize the list, maybe make a song up about them. But what we use here are these devices here that give you the first, you take the first letter of each of those, you can spell 3 different things. Private Tim Hall, so phenylalanine, valine, tryptophan, threonine, isoleucine, methionine, histidine, arginine, lysine, leucine. Or you can rearrange them, Private Matt Hill, or the one I use, Fill Empty Vat. So PHILLMTVAT is the one that I clinged on to when I was in your position. So if I remember Fill Empty Vat, those are the first letters. I just got to come up with the ones that are associated with those first letters. So those are the ones that we'll be balancing for in your lifetime. They're trying, but it's a challenge. So looking at protein, we already mentioned that crude protein is nitrogen times 6.25. This gets nothing about the amino acid or where they're coming from, how much ammonia is in the mix, how much nitrate or nitrite is in the mix. But we can go through and measure the nitrogen with a process called the Kjeldahl nitrogen. This is the original thing. It certainly moved us way forward in nutrition over the first 100 years it was used. I had the, I guess, terrifying experience of sort of running a Kjeldahl nitrogen. So what does that include? We take concentrated sulfuric acid, we put it in a Kjeldahl flask, a couple of boiling chips, and the feed sample. We put that on a burner. So we have basically boiling acid up on this rack in front of you. So the safety precautions are full face shield, full apron, that's acid resistance, because every once in a while those things will pop. So not something you want to have happen to your face. That Kjeldahl nitrogen, we have the decomposition of all the nitrogen down to ammonium sulfate. And then through a titration process, we boil everything off. We go into a known standard and we back titrate with kind of drops coming out of a burette. Certain number of drops means a certain number of, or certain amount of protein associated with that. So we need to know how much sample is going in there to begin with, how much nitrogen is generated, and we can back calculate this much nitrogen equals this much crude protein. We had this much dry matter to start with. We do crude protein over dry matter. That gives us a % dry matter for that. So we don't do things as nasty or as complicated. Everything tends to be correlated, but when all else fails, we have to go back to that original method. Another fraction we'll be talking about is what's called soluble protein. So instead of measuring all the protein in a sample as we do with crude protein, just like with NDF, insoluble versus soluble, we're interested in what fraction comes out when we extract it with a buffer solution, borate phosphate buffer, which is basically artificial spit. So how much of the nitrogen, or what fraction of the nitrogen, ends up in the soluble fluid part? So when the cow's chewing, how much of it comes out and is swallowed in the liquid form? So we balance for soluble protein in our ration balancing programs. We need a certain amount to make sure that we get optimum rumen crude protein or amino acid production in the rumen. during the fermentation. Sometimes we want to measure the ammonia. This is particularly important with our halages. Fermentations can go a little squiffy and turn a lot of the protein into ammonia. So ripping the protein molecules, ripping the amino acids apart, and what we're left is ammonia. If we have too much ammonia, we can overwhelm the system. One of the methods, there are several, to measure ammonia is called a colorimetric method, where we have an enzyme system that, proportional to the amount of ammonia, it'll produce a certain color. So the stronger the color, the more ammonia is present. So you're detecting that indirectly. You will have ADIN and NDIN. So we talked about this a little bit when we did our review of ration sheet or analysis sheets. So ADIN, acid detergent insoluble nitrogen. We have neutral detergent, NDIN, neutral detergent insoluble nitrogen. So this is the nitrogen associated with those two cell wall fractions. How much nitrogen is in the ADF fraction? How much nitrogen is in the neutral detergent fraction? So you generate the ADF, you measure the nitrogen through a Keldol or similar procedure. This is generally associated with nitrogen that is not available to the animal, ADIN. If you can't get it out with acid, boiling it with acid for an hour, the odds of the microbes in the rumen getting it out are pretty slim. So there's a certain amount of kind of default, 1%, 0.5% of the total nitrogen might be this damaged protein, but if we have some sort of heating during the silage making process, some sort of damage or bad fermentation, you'll get that caramel smell, or you get that halage that's black, or you'll get like a cigarette smell. Cows generally like to eat it, but the protein is not there, so they eat a whole lot, but milk production tanks because you don't have enough actual amino acids to support milk production. Some of the ADIN is available to the animal, and there are ration balancing equations that will address that. So protein in dairy cows, we measure the nitrogen, we multiply by 6.25 and we get that protein. We know that the microbes in the rumen can rearrange the protein for their needs. They're about 60–70% protein in their entire body. So they're going to take the amino acids that they have access to. They're going to use the ones they need. The ones they don't need, they'll rearrange, burn them for energy, rearrange them to the one amino acid that they need. So they can rearrange, they can build new protein, they can destroy protein, they can rearrange it, or they can completely ignore it as it goes through the rumen. And that's why it's so complicated or so hard to get our cows to amino acid level when we're feeding them. Knowing what goes on the rumen, figuring out from day to day, how does that all work, is a challenge. And it's a lot of probabilities, a lot of mechanistic approaches that have gotten us closer, but you still can't with perfect confidence, I feed this, this is what gets to the small intestines, because you don't know what's happening in the rumen. So balancing for protein, if we were to try on amino acid level like the pigs and the chickens do, we would be way off because of what happens by the microbes or what they do in the rumen. So we're getting better. We're defining just how much protein and what the profile is, how much of each amino acid are in particular rumen bugs going forward. So pigs and chickens, they do grams of amino acids that they need on a daily basis. They have it so dialed in that each week there's a slightly different proportion that meets what they need. But so the pigs and chickens, the essential amino acid is the focus. What happens in a more visual sense with our cows? So we'll introduce this and pick it up next time. So we're looking at it. We said that they can either build amino acids. So if you have the rumen, that's not working. If you have the rumen, so we have the rumen here, reticulum down here, we're looking at it from the left side. We have this creature here representing the whole entire population of the rumen bugs. They can build protein. They make it part of their bodies. They make new stuff. They can, how did I put that? They can destroy it. So what comes in as protein is ripped apart and no longer protein. They can alter it. They can take some of that protein, they can chew on it for a while. It falls out of their mouth. It's going to leave the rumen slightly different. The other thing, and we need to focus on the difference between the three we talked about and this last one, the protein just drifts around in the rumen, never gets touched, goes down to the abomasum small intestines where it is denatured, breaking apart by enzymes, and the amino acids are then released to the cow. And we'll talk about four different protein fractions based on what happens to that nitrogen. Is it ignored by the microbes or not? So when we get to this next week, so bring your hand out again. These two fractions, the A and B, are completely rearranged by the microbes. The C and D are completely ignored by the microbes. So, so we'll stop there, start next time. Did everybody sign our sale thing? Also, we need to make up a lab sometime this week. I think it's what, lab 5? So who needs to make up that lab, or whatever it was 2 weeks ago? So do you guys got your calendars or not? Okay, what about everybody else? Ready to make a decision as to when you can do it? Okay, so gather together and find an afternoon or an evening. I'm guessing that's your choices where it works. If anybody didn't sign the form here, please do so.
Is it just anybody that was involved, or
Did you do anything down there? Okay, anybody that's involved? So what would work out for, I guess, the 4 of you?
I do like tomorrow.
I have class at 3 o'clock. I think I have a meeting at 1, so that would be a challenge. What about next week or even the weekend? I'm available on the weekends. I don't know if you guys are or not. Okay, not this weekend, Monday. Next week?
Be after our lab.
Do you have vet or not? You have vet skills or not? Okay, so 3:30 next Monday. What do you got? 5 o'clock on Monday? Okay, 5 o'clock next Monday, we're making up, I forget which one, but we'll do it next 5 o'clock. I think it was 7, maybe?
Yeah.
7 or 6. Yeah, okay, so next Monday at 5, so we'll make that up then. Thank you.
Oh, yeah, I never saw you.
I didn't. I didn't, I need a pen.
Black.
I don't have a black pen.
I don't.
Okay, let me borrow it then. Okay, I signed it. Yeah, I did sign it. I signed it with whatever pen. Marvel signed it in blue.
I just want you to do it hot pink.
Bro, I should. I so should. I should. I should. Are you going to? Why don't you sign it? Hi. It's this. Lane, you want me to copy?
Not too dope, so.
Yeah, that's all you do during class is glue into your pen.
I want.
All you do